Have you ever pondered why is it that carbohydrates are so essential to our daily lives? Let's talk about carbohydrates, the unsung heroes of our diet. They're often seen as the villains in the world of nutrition, but in reality they play a crucial role in keeping us healthy and energized. They are a primary source of fuel for our bodies, providing the energy we need to keep our hearts beating, our brains thinking, and our muscles moving. But what exactly are carbohydrates? They're organic compounds made up of carbon, hydrogen, and oxygen atoms. The name carbohydrate is a hint to its composition. Carbo for carbon and hydrate for water, which is made of hydrogen and oxygen. Now, when we consume carbohydrates through foods like bread, rice, or fruits, our body breaks them down into simple sugars. These sugars then enter our bloodstream and are transported to our cells. Here, they are converted into energy through a process known as cellular respiration. But carbohydrates aren't just about providing energy. They also have other important roles in our bodies. They help regulate our blood glucose, prevent the breakdown of proteins for energy, and act as a source of dietary fiber, aiding digestion and contributing to a feeling of fullness. Moreover, carbohydrates serve as the backbone of our genetic material, DNA and RNA, and play a critical role in our immune system. They are also involved in cell-to-cell communication, a vital process for our survival. So as you can see, carbohydrates are not just about calories or weight gain. They are a vital component of our diet, playing a plethora of roles in our bodies. They are the fuel that keeps our bodies running, the key players in our body's functions, and the building blocks of our DNA. With this understanding of the role of carbohydrates, we will now delve into their complex chemistry, making it as simple as possible. Let's unravel the categories of carbohydrates, shall we? Carbohydrates, as you know, are organic compounds that are crucial for life on Earth. They are classified into three main types: monosaccharides, disaccharides, and polysaccharides. Firstly, we have monosaccharides. These are the simplest form of carbohydrates and are the building blocks for other types of carbs. Monosaccharides are composed of a single sugar unit and cannot be broken down into smaller pieces. Glucose, fructose, and galactose are prime examples of monosaccharides. Glucose, for instance, is the main source of energy for our body and is often referred to as blood sugar. Next, we have disaccharides, which are formed when two monosaccharides combine. The bond that joins these two sugar units is called a glycosidic linkage. Common examples of disaccharides include sucrose, lactose, and maltose. Sucrose, for example, is a combination of glucose and fructose and is the chemical name for table sugar. Lastly, we have polysaccharides, which are complex carbohydrates made up of more than two sugar units. These are typically used for storing energy or providing structure to cells. Starch, glycogen, and cellulose are examples of polysaccharides. Starch, found in foods like potatoes and rice, is a storage polysaccharide in plants, while glycogen is a storage polysaccharide in animals. So, in summary, carbohydrates can be as simple as a single sugar molecule or as complex as hundreds of sugar units bonded together. From providing energy to our bodies to giving structure to plant cell walls, carbohydrates play a pivotal role in the functioning of life on Earth. Now that we have our categories clear, let's explore how these carbohydrates are synthesized. How do these carbohydrates come to be in the first place, you may wonder? Well, let's explore this fascinating process together. The synthesis of carbohydrates primarily involves two key methods: the Kiliani-Fischer synthesis and rough degradation. Now, don't let these big names intimidate you. They're just pathways to creating the sugars we're all familiar with. Let's start with the Kiliani-Fischer synthesis. Named after German chemists, this method is like a magic trick that turns simple sugars into complex ones. Imagine you have a 5-carbon sugar, say arabinose. Want to make it a 6-carbon sugar like glucose or fructose? Well, the Kiliani-Fischer synthesis will make that happen through a series of reactions involving cyanide and acid. It's like adding an extra seat to the dinner table, accommodating one more carbon atom to join the party. On the other hand, we have the rough degradation. While Kiliani-Fischer is about building up, rough degradation is all about breaking down. It's like a reverse engineering process for sugars. So if you have a 6-carbon sugar like glucose or fructose, but you need a 5-carbon sugar like arabinose, rough degradation is the way to go. This process involves oxidation and reduction reactions that essentially remove a carbon atom from the sugar molecule. It's like taking away a seat from the dinner table, making room for a more intimate gathering. So in essence, these two methods work like a pair of biochemical sculptors, each with its own unique approach. The Kiliani-Fisher synthesis builds up, adding complexity to simpler sugars, while the rough degradation breaks down, simplifying more complex sugars. These methods give us the carbohydrates we need. But what happens when these sugars react? Well, stay tuned to find out more about the fascinating world of carbohydrate chemistry. What do you think happens when sugars react? Well, let's dive right in and explore the fascinating world of sugar reactions. To keep things simple, we'll focus on C5 and C6 sugars, namely arabinose, glucose, and fructose. These sugars are known to undergo a variety of reactions, but one of the most significant is the redox reaction, where sugars can be oxidized to produce aldehydes and carboxylic acids, or reduced to create sugar alcohols. This is crucial in many biological processes, including energy production and metabolism. Additionally, these sugars are capable of undergoing substitution reactions, where a hydroxyl group, OH, is replaced by another substituent. This ability to swap out parts of their structure allows sugars to form a vast array of different compounds, playing a major role in the diversity of life. But perhaps one of the most intriguing aspects of sugar chemistry is the phenomenon of mutarotation. Mutarotation, you might ask. What's that? Well, it's a process that occurs when a sugar in solution spontaneously changes from one isomeric form to another. If we take glucose as an example, it exists in both alpha and beta forms. The alpha form is where the hydroxyl group on the anomeric carbon is on the opposite side to the CH2OH group, and the beta form is where it's on the same side. In solution, glucose will spontaneously convert between these two forms, a process we call mutarotation. Why is this important? Mutarotation allows sugars to adopt the most stable form in a given environment, which can influence how they interact with other molecules. This is crucial in biological systems, where the structure of a molecule can significantly impact its function. So, through reactions and mutarotation, sugars can alter their structures to suit their needs, displaying a level of adaptability that is truly remarkable. These processes provide the foundation for the diverse roles sugars play in our bodies, from energy provision to cell signaling. These reactions and mutarotation processes are key to understanding the structures of glucose and fructose. Ever wondered about the structural intricacies of glucose and fructose? Let's dive into the fascinating world of these essential carbohydrates. Glucose, often referred to as dextrose, is a 6-carbon sugar, or hexose. It's a monosaccharide, the simplest form of sugar, and is the primary source of energy for our bodies. Structurally, glucose is an aldehyde with hydroxyl groups attached to each of the remaining carbon atoms. Its molecular formula is C6H12O6, and it exists primarily in a cyclic structure. Now, let's turn our attention to fructose. Fructose is another hexose sugar, but it's a ketone, not an aldehyde. It's often found in fruits, hence its name. The molecular formula of fructose is the same as glucose, C6H12O6, but its structure is different. It's arranged in a 5-membered ring, as opposed to the 6-membered ring of glucose. The intriguing part is that despite being composed of the same atoms, the different arrangements of these atoms in glucose and fructose result in distinct physical and chemical properties. For example, fructose is sweeter than glucose. In a nutshell, the structures of glucose and fructose are a testament to the beauty and complexity of organic chemistry. These simple sugars are the building blocks of more complex carbohydrates, and understanding their structures is fundamental to grasping the broader chemistry of life. Now you have a basic understanding of the chemistry behind the carbohydrates we consume daily. Carbohydrates aren't just about energy, they have medicinal importance too. Carbohydrates play a crucial role in the world of medicine. They feature prominently in certain drugs and treatments. For instance, they are used in vaccines to stimulate a better immune response. Some carbohydrate-based drugs are used to treat cancer, inflammation, and infectious diseases. They also play a role in wound healing and tissue regeneration. Carbohydrates are indeed more than just food. They are a crucial part of our health and wellness.